Sapsunthorn. Pakpoom received BS degree, double major in biology and computer science, two majors, in 2008. And he then got an MS degree in bioengineering at Stanford University in 2010. And currently he is a PhD candidate at the same place, bioengineering at Stanford University, working with Professor Drew Andy in the area of synthetic biology. Drew Andy is a professor in this area. He is considered one of the pioneers in synthetic biology. And today Pakpoom will give us a talk on his PhD research about thumb drive. See, I have one here. If you don't know thumb drive, this is thumb drive, a thumb drive. But it's not working on about this. His work is about biological thumb drive. It's a bio thumb drive using for storaging data. And to me, I consider this a very amazing field, amazing project, and quite interesting. I hope that you also find his work very interesting. It's going to basically give you a brief talk on what's been doing, as well as some basic in synthetic biology. And I think that's about it for introduction. Everybody, please welcome Mr. Pakpoom.
So can you hear me from the back? No. So I should use the microphone? Good. All right. So thank you again for having me here. So it's my pleasure to give a talk a little bit. I'll try to stay at the basic level about my research and also other exciting stuff in the field of synthetic biology. So before I get started, let me explicitly say my goal around this talk first. So before I came back home to Thailand, I talked to my advisor, Drew Andy, and discussed about a bunch of talks that I'm going to give in Thailand. And he asked me what do you want from that talk? You're going to spend some time talking to people in Thailand, and then what do you want from them? What do you want to happen? And after that discussion, so basically we came up with something. The first thing is in the short goal, if I'm successfully today, so basically I should be able to get you excited about the work. And maybe in the mid-range, we probably can figure out a way that we can work together and to lead the world in the long range. Okay? So this is my goal for today and also for other talks that I'm going to give in Thailand. Now let's get started. So if you think about biology, right? You have cell, and it gives rise to its progeny. The genetic material is passed down from generation to the next generation. And what happens is that you have some mutation that occur randomly in nature. There's some natural selection, and then it's passed on to the next generation. So because we, as a human being, are very smart, so we're figuring out a way to hack this process. So what we do is basically we took the natural living things like this, and we try to read out the information inside of it. For example, the genetic sequence that is inside the cell. And we keep that as the software or some digital information in the computer or something on a piece of paper. And we can use those information to write it back. For example, synthesize a new piece of DNA and put it back to a living thing so that we can create the living organisms that we want. All right, so basically, those are the big picture of biotechnology over the past several decades. So if you look at the first generation of biotechnology, here are what we have been doing. So we have recombinant DNA, we cut and paste the piece of DNA, right? We have the PCR to amplify the piece of DNA that we want. We have the things like DNA sequencing, so that we can read out what already exists in nature. Now, what about the second generation? So over the past decades, so here are things that on the process of developing and probably going to be the game-changing technology. So one is gene synthesis. So basically, we figure out a way to build the new piece of DNA that we want. And there are other stuff too. These two things are pretty vague concepts, but I can explain to you in the next few slides. So basically, it's the idea that we can put together a simple piece of DNA into a very complex device that can operate inside the living cell. All right. So let's see how this idea works. So let's suppose I want to build something inside the cell. Let's say I want to build a counter, a cell that can count a number, a cell that can remember, for example, like how many times it has been divided over the past, let's say, 10 or 20 days. Right? So I have this idea. And then I try to figure out a way that, in the next level, what are the piece of things that I want to put inside the cell so that I can assemble them together easily into the device like a counter. Right? And in the bottom left, and the next level below that, I try to figure out, like, in all of these basic devices, for example, you think about this as a counter. This is probably like, an individual bit, individual unit that can function as an information storage device inside the counter. And then you look at the next level, you can think of the genetic device as something that composed of the piece that can operate a simple function. Let's say a promoter, or like a gene, or the terminator, or like the ribosomal binding site. Right? And you look at the next level, basically to build these pieces, you want something that allows you to manipulate the physical pieces of DNA. So the idea of abstraction is that people who work in each of these different levels can interact with the people on the next level without the need to jump to the further level. So let me give you an analogy. So when you program on your computer, right, you don't actually need to understand all the complexity of the circuit inside the computer, as long as you understand the software. All right. On the other hand, the people who actually build the circuit inside the computer, they might not need to understand the physics of each transistor or resistor in the circuit. They only need to get that transistor or resistor that is working. Right? At the same time, the people who work on that transistor or resistor, they only need to interact directly to those who actually find the material to build those pieces. So basically, the abstraction idea is basically the idea that when you build something that's really complex, we can divide the tasks, and we don't have to go through all the detail in order to build something that is complex. But this idea hasn't been applied to biology yet. So one of the major goals of synthetic biology is to try to figure out a way to get this type of idea to work so that we can build something complex in biology. All right. So now, let me give you a specific example of this kind of research. So this is my research work that has been doing. It's ongoing work, it's not done yet. But I will show you some preliminary results that we just submitted a paper about like a few days ago. All right. So this is new, it hasn't been published yet. So the idea here is to build an information storage device that can operate inside the living cell. And let me tell you why it's important. So one application of information storage device that can operate inside the living cell is that it can be a tool that allows you to study the history of the cell. You think about application like when you study development or aging or let's say cancer, for example. Most of the basic biological questions that you ask is what has the cell been exposed in the past? Why this cell become cancer? And why this cell just stay as a normal cell? So why this cell become the brain cell and why this cell become a liver cell? So why this cell get older very quickly and why this cell stay young? Yes. 2, 3. 1, 2, 3. Thank you. So basically, like one of the possible application is that you can use it to study the history of the cell. Because most of the time, when you try to study the cell or the process that the cell has been through in the past, it's really hard to reach in real time. Maybe the cell is growing inside your body. Maybe the cell, the microbe that is in the environment. So if you want to get all those history, it would be easier if you have some kind of device that implant inside the cell and keep those history for you, so you can look at it at the end and refer back to what it has been through. For example, in this case that I draw the schematic picture here, it's basically the cell that can remember how many times it has been divided. Let's say, like, when you study development, one of the most important questions to know is what is the lineage history of the cell, right? Which cell developed to which cell. So if you have the tool that allow you to track all the lineage of the cell, that would revolutionize the way that we study development. Another important application is that you can use this memory as a tool to control complex behavior of the cell. So you can think of the computer without memory. It can't do anything much. And the reason is that most of the action that we need when we try to program the complex behavior inside any kind of machine, you require an ability to do a repetitive task. If you think about the programming, it's like when you write a for loop or while loop. Do this 3 times and then do that. Do this 5 times and then do those. So basically anything that allows you to order the cell to do that kind of thing, the cell that can divide 5 times, jump up and down twice, and then explode, for example, right? So all this kind of complex behavior also require memory, also require an ability that you have to keep track of what the cell has been through, all right? And I'm not talking about the memory that need to be compete with the memory inside the computer. When you talk about computer, you're talking about the memory that operate in the scale of, like, megahertz or whatever, right? Really fast memory, and the memory that has a really huge capacity in terms of like gigabyte or whatever. But this kind of technology, you don't have to go that far. You can go up to, let's say, 50 to 100 states. This scale is more than enough for you to track all the lineage, all the living thing on the Earth. So basically, like, we don't have to shoot that high compared to the silicon memory, but we need to get that kind of memory to operate inside the living cell. All right. Now let me show